Hello friends, welcome to semester 3. Today's topic of our discussion is structure of typical androecium and structure of tetrasporangiate anther. Before starting this session, let us know learning outcomes. Students, after listening this lecture, you will be able to understand what is androecium, then structure of typical stamen with suitable example and its function, and finally you will understand structure of tetrasporangiate anther. Students, androecium is essential whorl of flower. It is third whorl of flower. The member of androecium is known as stamen. Here in this illustration, you can see these all the members unite to form androecium and each member is known as stamen. So, androecium is usually made up of multiple stamens. In some plants, the stamens are sterile in nature. These stamens are treated as a staminose. The stamens are male reproductive organ. The typical stamen consists of filament, connective, and anther. The lower slender portion of stamen is called as filament, which is sterile in nature, while middle portion which connect anther and filament is called as connective, and the upper fertile portion of stamen is called as anther. The anther is a site of pollen development. There is great diversity in stamen number, size, anther shape, position, distance in the species to species. Let us know functions of stamen. The production of pollen is primary function of stamen. The pollen grain gives rise to male gametes during the process of double fertilization. They produce two male gametes. The sterile stamens, that is staminode, in some plants are the source of food for some insects, while petaloid stamen in canna attract the pollinators. So this is about functions of stamen. Let us know structure of tetrasporangiate anther. The anther is sac-like part of stamen which produce pollen grain or microspore. It is situated at the tip of filament. This is filament and here you can see the anther lobes. A typical anther is tetrasporangiate structure made up of two lobes and each lobe is two chambered. Here you can see two lobes of anther and this each lobe is two chambered. The two-chambered anther is called as dithecous. For example, citrus. In some plants, it is one-lobed known as monothecous or bisporangiate. For example, ricinus. This is the diagram of TS of young anther. The section is slightly four-lobed in outline. Every young anther in transverse section shows epidermis and archisporium. Here you can see the epidermis and this is archisporium. At corner of each anther, one or more archisporial initials are present. These initials undergoes periclinal division and forms sporogenous cells and outer parietal cell. The sporogenous cell act as microspore mother cell or pollen mother cell and gives rise to pollen grain. The anther wall consists of four layers, that is epidermis, then endothelium, followed by middle layers, and lastly that is tapetum. So this is a TS of mature anther. Here you can see that the tapetal cells are degenerating or disintegrating and the archisporial initial or pollen mother cell get metamorphosed into a tetrads and pollen grain. Let us see anther wall layers one by one. First of all, we will discuss epidermis. The epidermis is outermost and single layered with stretched and flattened cells. Here you can see these stretched and flattened cells. Sometimes these cells are lignified or cutinized. It is protective in function. The next layer, that is endothelium, which is present just below the epidermis. In general, it is single layered, but in coccinea, it is double layered. The cells of endothelium are radially elongated and shows fibrous bands. Usually, fibrous bands are U-shaped and they are hygroscopic in nature. These strands helps in dehiscence of anther. The layer of endothelium is discontinuous by line of dehiscence called as stomium. The next layer is middle layer. It is 1 to 3 layered structure just below the endothelium. These layers are ephemeral and get crushed during early meiosis in pollen mother cell. These layers persist in ranunculus and lilium. Sometimes these cells store starch. For example, in coccinea, they store starch. So this starch is utilized as a food by pollen mother cell during their development. Then our last layer, that is a tapetum. Here in this figure you can see these are tapetal layers. It is innermost layer of anther wall. It is a single layered and surrounds sporogenous tissue. These cells serves as a food material to sporogenous tissue. The tapetal cells are reddish brown pigmented in apple. There are two kinds of tapetum on the basis of their behavior, that is Amyboid Tabitum and Glandular Tapatum. Let us see Amyboid Tabitum. It is also called as periplasmodial type and considered as a primitive one. Due to the action of hydrolytic enzyme, inner and radial walls of tapetal cells break down. Their protoplast penetrate between the pollen mother cell and developing pollen grain. There is a development of mass of tapetal periplasmodium due to fusion with each other. This tapetal periplasmodium remains associated with the pollen grain up to their maturity. As the anther gets drying up, the tapetal periplasmodium gets dehydrated and coated over the surface of pollen grain and forms exine. Such a kind of amoeboid tapetum is found in lower monocots like Tradescantia, Typha, Sagittaria and Potamogeton. The next type of tapetum is a glandular tapetum. It is also called as a secretory tapetum. The cells of this type of tapetum secrete some substances from their inner faces and the end of meiotic division in the pollen mother cell. The secretory tapetal cells are thin and contain cell organelles. The tapetal cell forms spherical proubish bodies within the cytoplasm. In many plants, proubish bodies are originate from endoplasmic reticulum of secretory tapetal cells. These proubish bodies pass out through tapetal cell membrane and coated with sporopollenin. These bodies are called Ubish bodies, which are responsible for thickening of exine. Such a kind of tapetum is found in higher monocots and many dicots. For example, sorghum, avena, citrus, capsicum. Now we will discuss functions of tapetum. The tapetum is involved in the supply of nutrient and food to the developing pollen grain inside anther. The tapetum plays a role in the breakdown of callose wall around the pollen tetrad by secreting an enzyme callase. The tapetum plays an important role in the formation of exine. The pollen kit is also formed by tapetal layer, which is an insect attractant and protect pollen grain from ultraviolet light. So these are the functions of tapetum. Students, I hope with the help of this presentation, you have understood what is androecium, then structure of typical stamen and structure of tetrasporangiate anther. Thank you for listening me.